And the way that I structured it, I'll email you all the handout that I'm kind of using to guide the talk, is just asking 3 basic questions about each medication. Why should it work? Does it work? And should we use it? The first drug, Vortioxetine or Brintellix. The question of why should we use it? No, that's not the first question. The first question is why should it work? So it actually has a cool pharmacodynamic profile. There's this pseudo-new class called the serotonin modulators, and vilazodone is in that class. So like vilazodone, it's able to bind to the serotonin reuptake transporter, but not to saturate all of them. So most SSRIs will saturate at about 80%, and it's thought that that kind of massive serotonergic effect is correlated with a lot of the side effects, the sexual side effects and other side effects that can afflict many people who take SSRIs. So what's kind of cool about the mechanism for vortioxetine, even though it's not truly a novel mechanism or a novel drug, is that at about 40% of saturation of those serotonin transporters, you're actually able to derive an antidepressant effect in both animal and human models. In addition to blocking the serotonin transporter and having partial agonist activity at 5-HT1, like Buspar and like vilazodone, it also antagonizes this 5-HT7 receptor. 5-HT7 is like pretty cool right now in the literature. I actually found a more compelling explanation or justification of the 5-HT7 hype when I was looking at this third drug, Latuda. For Latuda to work as an antidepressant in animal models, 5-HT7 has to be intact. So when they do knockout mice, 5-HT7 knockout mice with Latuda or lorazepam, you don't replicate the antidepressant effects in animal models. And I thought that was like pretty legit because they're like 12, 14 serotonin receptors, and we're always busting them out, 5-HT3, 5-HT6, 5-HT11, and we don't really know what they do, and we don't really understand the serotonin system. So I was skeptical of 5-HT7, but right now I'm kind of liking it. So in addition to those pharmacodynamic properties, it has some elements of dirtiness. So it does enhance serotonin, norepinephrine, dopamine, acetylcholine, and histamine models, at least in animal histamine levels, at least in animal models. And then jumping to this next question of does it work? I think it does. So it was approved by the FDA based on 6 large studies. It's been approved at a dose range of 5 to 20 milligrams. And its record is not bad. So it's 3 and 3 wins and 3 losses. That may seem like not that great, but in clinical trials, given the historic and astronomical rise of the placebo effect, it's not bad to defeat placebo in 3 trials. The other cool thing about these trials is that in the 3 positive studies, there were robust separations between drug and placebo on the MADRS. So you were getting like 4 to 6 point separations. And also on the Hamilton. And the critical study in 2011, I think, by Kirsch, when he claimed that all antidepressants don't really do much, that was based on the FDA's general approval level, which if you look at it for all the other antidepressants, the separation on the Hamilton had been 2 points. So 4 to 6 points is something. It means something. And also we're talking about separation in rates of response and remission of 15 to 20% versus placebo. So it seemed to be working in those 3 positive studies. The other cool thing about the literature around this drug is that the pharmaceutical company had the fortitude to test it against active comparators. So they tested it against Cymbalta and they tested it against Effexor. They did not have the fortitude to do a formal statistical comparison of how it did compared to those agents relative to placebo. Was it actually better than Effexor, or was it actually better than Cymbalta? If you look at the trends, it does seem that the SNRIs are slightly superior. Particularly Cymbalta looked like it did a little better. But I thought it was cool that they threw those drugs in there and that they showed that both the Cymbalta or the venlafaxine and this new drug Vortioxetine work. Third question, should you use it? So I'll just go through awesome attributes and attributes that aren't so awesome. And then you can decide if you want to use it. This is really, I think, the breakthrough for the drug. It is super well tolerated. So the rate of sexual dysfunction in these large studies, we're talking like N's of 500, was 1% or less. It's like placebo. So maybe better than placebo. It was just well tolerated. So the most common side effects reported were nausea, headache, and dizziness. Another very cool feature of this drug, the graphs look beautiful in the studies. I mean, they look so good that you're like, really? Could this be an actual study? What did they do to make it look so good? But this drug separates from placebo at 2 weeks in one study and at 1 week in another study. So there's evidence that it works faster than our other antidepressants. And then the other thing that convinces me that this is a worthy drug is that if you look at the breakdown by subscale, so the MADRS is composed of these 10 items that measure different dimensions of mood. And it can be easy sometimes to manipulate a study, if you have Seroquel, for example, in an antidepressant study, and Seroquel knocks everyone out. And that's a heavily weighted item on a depression rating. It can look like it's having a really awesome antidepressant effect, but really you're just knocking people out. So if you look at the distribution of effects among subscales, it's 9 out of 10. Yeah, 9 out of 10 items on the MADRS separated from placebo. So the MADRS is very cool for those of you who haven't used it. It has these sort of poetic items like lassitude, sadness, inner tension. But it does seem to be working across domains. And then it's got nice pharmacokinetics. It's kind of like Lexapro, no drug-drug interactions. It's got a lovely half-life of 6 to 7 hours, so probably unlikely to cause really acute withdrawal symptoms if people stopped it. It might be easy to taper. And then there's this idea that it may be more effective for mixed anxiety and depression because like Buspar has that 5-HT1 partial agonism. All right, what's a bummer about this drug? So multi-centered studies in Europe, Asia, and Africa. These studies were not done in the United States. And I think a lot of us in the clinical trials world believe that the studies that are done in other countries may have lower quality. So that's kind of a bummer. Also, you always kind of worry when I think one of the studies had involved 68 countries. You're like, really? How do you coordinate a study across 68 countries? There's just so much potential for mishap and error. Another thing that's not so cool is that it did not seem to, I may have mentioned this already, but it did not seem to be as effective as the SNRIs. Again, they didn't do a formal analysis. And then, like most antidepressant studies, these are not our patients. So these were patients that didn't have comorbidities, they didn't have substance abuse, they didn't have suicidality. That was really concerning.